This is part F, control of blood flow. Be sure and try to complete the previous parts for continuing. Blood flow to the different organs varies depending on their needs. We'll discuss pressure and resistance. This is partly a review from what we've already covered. We'll look at control of blood flow and then central or reflex control of blood flow. Pressure. Remember that an increase in pressure difference equals an increase in flow. Pressure differences are determined by the pressure generated and the resistance. Blood pressure generates pressure. Blood pressure is determined by blood volume and arteriolar resistance. Blood pressure is measured using systolic and diastolic. For example, 120/80. Systolic pressure occurs during systole, and the force is the contraction of the heart. Diastolic pressure occurs during diastole. What's the force causing diastolic pressure? If you think relaxation of the heart, oh no, that's not it. How can relaxation generate pressure? It has to be something pushing. During systole, blood is moving into the arteries. When that happens, the walls of the arteries expand because they're elastic. And during diastole, the walls recoil, generating the pressure and forcing the blood out. So the force moving the blood during diastole is the elastic recoil of the arterial wall. Pulse pressure is the difference between systolic and diastolic pressure. What do we mean by blood pressure? Well, here's a profile of the arteries, arterioles, capillaries, and veins in the cardiovascular system. The arteries are shown first, then the arterioles, the capillaries, and the veins. The chart shows blood pressure. Notice the blood pressure drops slightly in the arteries, but it's essentially the same. We refer to the arteries as the arterial tree because it's arranged something like the trunk of a tree. The aorta and then all the branches would be the branches of the tree. The pressure decreases continuously from arteries to veins. After all, fluids flow from high to low. If the pressure didn't decrease, the blood would flow backwards. Compare the pressure at the distal end of the veins with the pressure in the right atrium at the beginning of the cardiac cycle. In the distal end of the veins, pressure is, maybe 15. It's around 0 at the beginning of the cardiac cycle. The pressure in the veins is slightly greater, therefore, than the pressure in the right atrium. It has to be, so the blood can flow from high to low, from the veins into the atrium. Mean arterial pressure is the average pressure in the arteries. Now, not shown in the graph is the fluctuation. In the arteries, the blood fluctuates between 120 and 80 as the heart beats. Also shown in the chart is the velocity of blood flow. Notice that the velocity is high in the arteries, low in the arterioles and especially the capillaries, and high again in the veins. The total cross-section area is also shown. It's lowest in the arteries and veins and highest in the capillaries. Now, that may not be intuitive because you're thinking, well, arteries are large, so are veins, capillaries are small. How can the total cross-section area be greater? Well, as the cross-section area increases, the velocity decreases. Here's a view. Let's say the big circle represents the total cross-section area of many small vessels. It's greater, quite a bit greater than the total cross-section area of the one big vessel shown on the right. Imagine you're sitting next to a stream and you're watching a leaf flow down the stream. When the stream narrows, the flow speeds up and the leaf rushes through the narrowed part of the stream. And when the stream widens again, the flow slows down. Well, that's the same thing with the blood. Where the arteries have a low cross-section area, the flow is fast. Where the vessels have a high cross-section area, the flow is slow. Resistance. The function of resistance is to control, that is, vary the blood flow to different organs. Hyperemia is increased blood flow. Ischemia is decreased blood flow. A decrease in resistance equals an increase in flow. Resistance is determined by viscosity of the blood, length of the vessel, and diameter or radius of the vessel. We've covered this already, but we're reviewing. Now, one of these is used to determine the blood flow to the various organs of the body. It's done by varying resistance. Would the viscosity of the blood vary from vessel to vessel? No, the viscosity of the blood is the same throughout the system. What about the length of the vessel? Would a vessel elongate to increase its resistance and shorten to decrease its resistance? No, the vessel length is the same for a given vessel. What about the diameter of the vessel? The diameter does change. Remember, circular smooth muscle, when it contracts, the vessel constricts. If radius is doubled, the flow increases by 16 times. Therefore, a slight change in diameter results in a great change in flow. Which of these would control resistance in the cardiovascular system? Well, we just went over that. It's the diameter. Arteriolar diameter is the chief determinant of resistance. The design of the cardiovascular system will help you understand what we're getting at. The design is not as the one shown in the diagram. The vessels are not aligned in series. In other words, the heart does not pump blood through one set of capillaries and then through another set of capillaries in a different organ and then through another set in still a different organ. Instead, the correct design of the vessels is in parallel. Now, this is a simplified look at the branches of the cardiovascular system. Notice the heart pumps blood through a single vessel, arteries. The vessel branches into three, and then the blood travels through capillaries of certain parts of the body. The three main branches are the skeletal muscles, the vital organs such as the brain and the heart, and the other organs of the body. Well, what about the resistance? What happens when it changes? If there's a constriction in the arterioles at number 5, what happens to blood pressure at number 2? Well, the constriction hinders the flow of blood. Pressure would build up before the constriction, so the pressure increases. What would happen to the blood pressure at 8? Well, the constriction hinders the flow of blood beyond it, so the pressure decreases. This may be intuitive to you, thinking of, say, spraying water out of a hose. What about the pressure at number 1? Well, if 5 constricts, the pressure at number 1 will increase, not as much as it did at point number 2. Another way to look at it, if there's a constriction at point 5, what happens to the blood flow at 8? Now, this is more intuitive. You constrict 5 and you would decrease the flow beyond the constriction. What about the blood flow at 9? Well, you're restricting flow at point 8, so the blood has to go somewhere, and so that would increase the flow through point 9. What about point 10? Well, it would also increase the flow through that point. Imagine I have a tube hooked up to a faucet, and I have a Y-shaped connector on the tube, so it branches into a left and right branch. I turn on the water so it's flowing out of the tube. If I squeeze the left branch, the flow decreases through that tube, but it increases through the other tube. If I squeeze the right tube, the flow decreases through that one, but it increases through the left. If number 7 constricts, what happens to the blood pressure and blood flow in other parts of the system? The blood pressure increases and blood flow increases. The system, you'll note, has pressure detectors. What's the function of these pressure detectors? Well, obviously, to monitor the blood pressure. Why is the pressure detector located where it is? To monitor blood pressure to the vessels leading to the vital organs. Peripheral resistance is the resistance in the systemic circuit. Pulmonary resistance is the resistance in the pulmonary circuit. Two types of control of resistance are local control. This is control not involving nerves or hormones, and it acts in a localized place. And central control. This is control that involves nerves or hormones. It's also called extrinsic or reflex control. We'll talk about both of these. Let's look first at local control. Its function is to regulate blood flow to specific organs. It's especially important in the muscular organs, such as the heart, skeletal muscles, and GI tract. There are 4 types of local control. One is called metabolic vasodilation. As you'll see, this is a very important function. Another is called flow autoregulation, and a third, reactive hyperemia. The fourth is inflammation. Let's look at metabolic vasodilation. This is sometimes called active hyperemia. Hyper means greater, and emia comes from heme, meaning blood. When the activity of an organ increases, say your skeletal muscles begin to increase their work, changes result. For example, there's a decrease in oxygen because the organs are using up the oxygen. There's an increase in carbon dioxide because the organs are generating that as a waste material. There's an increase in hydrogen ion concentration. You'll understand why later. Carbon dioxide mixes with water to create an acid. There's an increase in metabolic intermediates, an increase in the secretion of prostaglandins, signal molecules, an increase in kinin secretion, another type of signal molecule. There's also an increase in another hormone called nitric oxide. All of these changes relax arterial or smooth muscle, and when that happens, the arterioles dilate. Why do they dilate? Because the blood pressure within them forces the walls outward. The organ receives increased blood. About flow autoregulation. An increase in systemic blood pressure contracts arterial or smooth muscle, and it constricts the arterioles. That's a way of negative feedback to regulate the flow to a specific organ. So it maintains a constant blood flow to the organ. Reactive hyperemia, if the blood supply to an organ is completely stopped and the blockage is removed, there's a great increase in flow, higher than would be expected by just restoring the flow to normal. Inflammation is a response to injury and infection. We've talked about this already. Chemical mediators such as histamine are released from the injured tissue, arteriolar smooth muscle relaxes, and the arteriole dilates. What about central control? The central control has a different function. Its function is to maintain overall blood pressure to provide adequate blood flow to the vital organs, such as the brain, the heart. It includes sympathetic nervous system, parasympathetic nervous system, and epinephrine. There are other factors that influence the overall control. ADH, for example, also called vasopressin, vessel, pressin, pressure. Angiotensin is another, and atriopeptin is another. Let's look at the sympathetic input to the vessels. It includes vasoconstrictors. These are also referred to as vasomotor nerve fibers. They arise from the vasomotor center in the medulla and the pons. They increase sympathetic input to the vessels, which causes constriction of the vessel. They function by releasing norepinephrine to stimulate alpha adrenergic receptors to contract the smooth muscle. This is very important in the arterioles, so much so that when we mention sympathetic input to the vessels, we're talking about vasoconstriction. They provide something called vasomotor tone. Now remember, once again, control systems are like volume switches, and they can be turned up or down rather than on or off. The vasomotor tone regulates the state of contraction or dilation or constriction of the vessel. There are some vasodilators in the sympathetic nervous system, but these are not so important in blood pressure control. They're found in some skeletal muscle fibers. They involve beta 2 receptors and sweat gland arterioles. They release acetylcholine or norepinephrine. They function in exercise and stress. They function in sort of an anticipatory response before exercise begins. They function locally, but they're central in origin, so we place them under control of blood flow from the sympathetic or the central control. The parasympathetic input to the vessels is almost non-existent, very few. They dilate vessels in the external genital organs and the GI tract. For example, they promote the erection of the penis. They function by secreting nitric oxide to relax smooth muscle. Epinephrine plays an important role. It acts on alpha adrenergic receptors that control vascular smooth muscle to constrict vessels in organs not involved in stress, such as abdominal visceral organs and skin in heavy exercise. In light exercise, skin arterioles dilate to radiate heat. Now, what's going on here? By contracting the vessels and constricting the flow to these organs, blood is made available for the vital organs involved in stressful activities like the skeletal muscles. As a result, blood flow to skeletal muscles is increased. It acts also on beta 2 adrenergic receptors that relax vascular smooth muscle to dilate vessels. These are the vessels that are involved in stressful activities such as skeletal muscle and cardiac muscle. Before we move on to ADH, notice that epinephrine has differing functions depending on the type of receptors that they act on. And once again, to repeat, it's a way of shunting the blood flow from the less vital organs to the ones involved in stressful activities. Well, what about ADH? You heard me mention this briefly when we introduced the endocrine system, and I said we'd come back to it later. It's secreted by the posterior pituitary, and it constricts vessels. It's not important in day-to-day regulation. It's more involved in long-term regulation. And we'll look back at this when we get to the urinary system. Another is the angiotensin system. Now you see the diagram, and yes, it looks complex, and it is. It's a complex system involving many organs of the body. Angiotensinogen, it's an inactive form, is secreted by the liver, and it's activated in two steps by renin from the kidneys, and it generates angiotensin 1, and ACE, angiotensin converting enzyme, from the lungs and kidney to generate angiotensin 2. This is the active form. It acts on its target, systemic arterioles. The response to the constriction or is constriction of the vessels, and it raises blood pressure. Now, remember the scheme we use when we try to understand endocrine function. We look at the gland, we look at the stimulus, we look at the target, and the action of the target. In this case, it's a little complex. The target are the systemic arterioles, and the action is to constrict. The hormone is angiotensin 2, and I guess you could say the source of the hormone ultimately is the liver. Another hormone called atriopeptin, also called atrial natriuretic factor, or ANP, dilates vessels. It increases salt and water excretion, and we'll come back to this when we get to the urinary system. It's involved in long-term regulation of blood pressure. This is the end of part F. We're now ready to continue with parts G and H under the cardiovascular physiology.